She got lymphoma. She survived, but you know, the treatment for cancer is just as bad or worse than disease, and that really like put a chip on my shoulder that and kind of frankly disturbed me in a way that I had never been able to shake. So that you know, that really drives me. I've talked to thousands of people about genetics and science, and I think sometimes when it hits home for you, a lot of common objections go out the window because it's very real. You know, when people get sick or you lose people who you love. Yeah, my mom actually passed away of cancer during my PhD during COVID. I was unable to visit her because of quarantine. And I think I got about 2 hours with her before she passed. So that actually really just strengthened my resolve to pursue this mission. Anyway, so I work at minicircle, and today I'm going to talk about, I guess this could probably be broken into like 3 categories. So, the term Devon and I use for our collaboration is Project Limitless, which is up there. And Devon, I drew I made an AI image of like a 50-foot tall angel with like a sword, like walking over a city in ruins for like a music playlist I made Devon, and he really responded to that. And I thought I was hitting the right tone. And then he actually drew on a piece of cardboard in his house an even better image. It was the planet Earth with an angel standing on top of Earth, pointing a sword into space, and DNA from every continent flowing up into the body of the angel, and it said, "Together farther." So Devon always like undersells his background and experience. Like, I would say he has roots in some of like the darkest corners of the human experience, but he's endlessly capable of overcoming that and striving for good and reaching for something higher, which is super inspiring. And I think Together Farther and that image really encapsulates that. So this talk is going to be just like DNA 101. So many people I've talked to, adults, like well-educated adults, do know that DNA is like, looks like this kind of, but that's about where it ends for many people. And the truth is, DNA is not that complicated. One of my big goals tonight is to explain DNA to everybody here in a way that you can take home a fairly advanced, actionable understanding of DNA and human genetics. So that's the first part of the talk. The second talk will be, we'll talk about sequencing. So let's say DNA sequencing, and we can call it like whole genome sequencing. This is a lot of what Devon and I collaborate around. We've been traveling the world sequencing like elite performers, mostly from strength and like intellectual fields so far, but it's been super productive. And 3, I'll talk about genetic engineering. Genetic, gene therapy or genetic engineering, however you want to say it. So yeah, this is going to be the talk. So DNA 101. Can anybody tell me what DNA is?
Yeah,
Good. Go for it.
Blueprint
That's a very good answer. What's a blueprint for?
for your entire makeup of your body.
Very good. Blueprint is a key word. So I agree strongly with blueprint. Does anybody want to add anything specific to that?
it codes for proteins.
Very good, Auden. So Auden, like his dad, is a very fast learner. So DNA encodes for proteins. What are proteins? Anybody?
Amino acid chains.
Amino acids, they are chains of amino acids.
Building blocks.
Building blocks, that is fairly accurate. Anybody else? Like what are proteins in the context of cells, in the context of biology?
Information. Structure?
Structure, and for DNA is more like the information. Proteins are more like structure. Anything else? So proteins are machines. So all Earth life, anything that's a machine, it can be structural, it can be moving, that is proteins. And DNA is the instruction, like the blueprints for all the machines. So when you get to molecular biology in college, this is like one of the first things they teach you. DNA, one-way arrow, proteins. Like that's super important. So in the context of a cell, how am I going to do this? I think I'm going to do it like this. So we all know this might be too simple, but human body is roughly like this. If you're Devon, it was like
Just the one arm. Just the one.
Yep. And then life is made of cells. Does anybody know how many cells each of our human bodies is made of, approximately? How many?
3 trillion.
Trillion, 3 trillion. That's correct order of magnitude, more or less. Very good guesses. I'd say about 10 trillion. So those are big numbers. Anybody have an idea of the scale of that number? Can anybody have any thoughts about how big is 10 trillion?
Seconds would be like 30,000 years.
Let's see, so if you count a number a second, never sleep, it would take you 11 days to count to 1 million, 33 years to count to 1 billion, 33,000 years to count to 1 trillion, 330,000 years to count to 10 trillion. And so keep in mind that ancient human history is 2,000 years ago. So 10 trillion is an unthinkably huge number, and that's cells. So that's how many cells we have in our bodies. Yeah, I'm pretty interested bigger.
Yeah, is that just our cells?
Great question.
Just our cells.
So, first of all, this number, I'm actually like lowballing this estimate. It's probably like 50 trillion or something, but a lot of those are red blood cells, which we're not really going to count. You actually have about 100 trillion microbes in and on your body. Mostly, we're like a tube in a tube, and the gut is the inner tube. Inside the gut, you have your gut microbiome, and then on your skin you have microbiome. There's about 100 trillion of those cells. So that's a whole layer of complexity that science is really struggling to decipher and make actionable, but unthinkable numbers. Each cell is basically as complex as New York City. It has a downtown, which is like the nucleus, and then there's power plants and highways and all this stuff. It's honestly amazing that we work at all biologically. At the same time, we do have legitimate gripes about some dysfunctions in our biology, but so we have 10 trillion cells, and DNA is in the middle. It encodes proteins, which are machines. Proteins, we see that all the time in food. What other molecules are talked about in terms of food and macronutrients? Anybody?
Alcohol.
Alcohol is a critical food group. Any others?
Carbohydrates.
Carbs. Okay.
Phospholipids.
Phospholipids would go under fats, but fats and carbs for sure. Anything else? Any other major dietary components?
Fiber.
Fiber's a good one. Water, electrolytes are two major ones. Vitamins and minerals. I would say this is like a great way to simplify cell biology. So fats and carbs are energy to fuel the proteins. The cell is filled with water and salt, and the membrane is made of fat. DNA, fat can be structure in the membrane, or it can be energy to fuel protein machines. Carbs are just energy, they're not really structure. It's filled with water and salt, and DNA is the blueprints for all the machines that actually do anything. Does anybody have any questions or thoughts about that? Okay, there's a human body, about 10 trillion cells. This is more or less how each cell works. In terms of this DNA, does every cell have all our DNA? How much DNA is there? Does anybody have any ideas about this?
Every cell is a whole
Only cell with
DNA.
nucleus.
Was it?
Only cell with nucleus.
So only cells with nuclei, that's correct. Yes, every cell does have all of our DNA. Does anybody know how much DNA we have?
Is it 3 billion lines of code in there?
3 billion digits, yeah.
Yeah.
Basically, yes. I wouldn't say lines, I would say digits. So there's 3 billion digits of DNA code. Computer code is like zeros and ones. Our human alphabet is 26 letters. Anybody know what the options are for DNA code?
A, C, G, T.
Different cells in chromosome.
ECTG. I know Devon is a fan of the movie Gattaca. The movie is spelled only, the title is spelled only using the letters of DNA code. G's, A's, T's, and C's.
Did they do that on purpose?
They absolutely did.
That's amazing.
So that's an easy way to remember it if you're having trouble remembering. So we have these 3 billion digits of DNA code. The entire 3 billion code is in every one of our 10 trillion cells, which is kind of insane, but that's the way biology works. So how is this code organized and what does it do? Our DNA is basically like a book. It has It has 23 pages, and each one that's like a chromosome. As scientists, we round numbers a lot. If there's 3 billion digits and about 20 pages, that's about 100 million digits of DNA per page. So now, there's 23 pages in the book. The book itself is called the genome. That's a word that I think most people have heard, but not a lot of people maybe know exactly what that word is. Does anybody know what a gene is?
So your trait passed on from
What's that?
a trait passed on from your parents, like a gene,
That gets close. That gets at the nature of a gene and genetic inheritance. I'm looking for a more specific definition. Anybody? Any ideas?
Is the area in your genome that codes for a specific protein?
That's excellent. Yes, that's definitely. Yes. So a gene, let's say it'll be like blah 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 blah, GTC, something like this. This would be a gene, and it makes one protein. So this begs the question: so a protein is a machine. How many machine parts do we have in our library as humans? How many different options do we have? How many can we make?
Good question. A little bit different.
Okay, yeah, what's your question?
What's the importance of understanding this? Like, why understand this right now?
Because it helps speak to the importance of sequencing and why getting your sequencing data, that data is going to be more interpretable than most people realize. The two big objections to sequencing are 23andMe stole everybody's data. You can easily get sequenced anonymously, so that's very quick fix. The other one is, what am I going to do with that data? And that's more valid. Devon, so in collaboration with Devon, we've developed a pipeline through like a year and a half of work. We can analyze your whole genome in like 30 to 60 minutes. And that'll be one of the things you can DM us or something at the end, and we're basically happy to loop you into that process. But the other objection is, so it's mostly to make sequencing and gene therapy more tangible for people. The genome is just very abstract. You see the double helix, you hear as huge numbers. But, making it like this, connecting it to stuff we eat, hopefully it makes it more tangible for everybody. Yeah.
Thanks.
Yeah. So how many options do we have for protein machines? Anybody?
It's about 2,000?
2,000?
Yeah, about.
First question on and got
Oh, no.
it slightly wrong. About 20,000. So we have about 20,000. This book's about 23 pages. That's about 1,000 per page. So at this point, you'll notice we've gone from 3 billion digits of code, which is like intractable, to now we have a 20-page book, and there's 1,000 words per page. Somebody gave you a 20-page book. That's easy to read. That's a short book. So yeah, this is basically to say that this is more tractable than people might realize. So 1,000 genes per chromosome. Does every person have the same genes? No? Anybody who thinks yes? Some people do? What? You think yes?
I think yes.
I'm going to ask you to refrain from answering.
Okay.
You've been listening way too close, so you know basically every answer. Every human has the same genes. We all have the same genes. We all have the same book. Every human. So yeah, Eric, again, to touch on your question, like I've had these conversations with like hundreds of people. These issues come up so much. Like, do I have the gene for intelligence? I'm like, we all have the same genes. So this is to hopefully start spreading easily accessible basic understanding of genetics. Yeah. Every human has the same genes. What's different then if we have the same genes?
Some of them are turned on and some of them are deactivated.
Yes, that's right. So, would you know? Does anybody know how that's accomplished?
I do not.
Okay. How about you?
How turning it on is accomplished?
Yeah. How do you turn on and off?
I mean,
Experiences.
And what is-
environment.
Yes, but what is the molecular mechanism through which the environment impacts which genes are on?
Proteins?
Proteins are, if it gets done in a cell, it's done by a protein. But proteins use tools. There's a popular term these days.
Epigenetics.
Epigenetics, yeah, epigenetics. Epigenetics is like, so notes in the margin. And it should be black. Hopefully this gives it like this. If you put like something like that, notes in the margin are epigenetics. That tells you like what parts of the book to pay attention to. There's one other important element that makes people's genes, so that doesn't make your genome, your book different. That just makes how your cells read the book different. We all have slightly different copies of the book in us. What is the nature of our differences in our books in this room, in all of humanity? There's probably a word that most of you know and would use for this. Like, what are the X-Men?
mutation.
Mutants, yes, we're all mutants. The scientific term for it is variants, but these are like typos in the book. Okay, so the biggest source of mutations is making copies of DNA. So you start life as one cell and you turn into 10 trillion. Every time that cell divides, that's the biggest source of mutations. More than smoking, more than the sun, more than anything. We each have about 5 million mutations per person. So we all have the same book, we all have the same genes. We each have a unique set of 5 million typos in our genome. If you randomly make typos in a book, how likely is it that it makes the book better?
Not really.
Not likely. Yes. Most mutations are bad. Some are good, but most are bad. And The only thing that like keeps good ones around is did it get you to baby making age. After that, it doesn't really matter if it kills you slowly. So the shape of a human life is basically you're in really good shape till 25, and then it's time to die slowly. That's how it works. There's a hard mathematical limit on 120 years in the human lifespan. Anyway, so this is how DNA works. It's also worth noting, these words are very spaced out. Only 1% of the book is words. The rest of it is other stuff. I'm not going to go into all that, but I will say a lot of the other stuff, like maybe 30+%, is literally just DNA sequences inserted by viral invaders over the course of millions of years. Martin, feel free to tell me I'm saying this wrong.
No, you're absolutely
Okay.
correct.
Yeah, so, you know, Devon probably has, in fact, I know he has exceptional genetics in many ways. But Devon and all other humans, our baseline, good or bad genetics, is 30% of our genome is just viral invader leftovers. This is not good, okay? So when our starting point is 30% viral invaders and 5 million typos per person, that is why these are so important, okay? Nature is miraculous, but does not care, okay? It doesn't. So, not about the same things we care about exactly. Anyway, so this is what DNA is. DNA is code for proteins. We have a library of 20,000 proteins. Should be on here somewhere. So DNA encodes proteins. We have 20,000 proteins, about 1,000 per page, about 1,000 per chromosome. 3 billion digits total, 5 million mutations, so about 1 in 1,000 bases is mutated. Does anybody have any questions about any of this?
So many questions. but it's good.
Yeah, so one way that this helped me, Eric, again to speak to your question. So at our company, minicircle, we make gene therapy. Our first commercially available product, follistatin, the packaging is pictured right there. We sit around and brainstorm, what's the next gene therapy we should make? And through collaborating with Devon, I came to this 20-page book idea, and I literally, in Excel, got all 20,000 human gene names, organized them, 100 rows, 10 columns per page in alphabetical order, and I made literally a 20-page book with all 20,000 genes, and I would just read through it and highlight ones I know, highlight ones I don't know. I learned so many genes and so many facts about the genome that way by making it a physical, like, object that I could interact with instead of just some abstract thing. So yeah, at the end I'll give you some social media to follow, upgrade, minicircle, Devon, me, all these things. Some of those socials will have some educational material like this book and other things like this. Anyway, so that's DNA. It makes proteins, 20,000 proteins. We all have a bunch of mutations and viral leftovers, and that's generally not a good thing. So who here thinks with that information, or just based on their life experience, they might be interested in, like, who would want a better genome? Who's awesome. Zach's genome is already perfect. Zach personally, he is awesome. So the first step towards, might as well put .com there. They're not an official partner, but like we've done all kinds of work with sequencing.com. So the first step towards, let's say, human mastery of genetics, like we want to be a spacefaring civilization. We also don't want people to be born with like terrible ravaging disease. We don't want all kinds of things. Genetics is necessary for this. So I will tell you that, little history of genetics, 1955 we discovered the structure of DNA. Gene therapy became a thing maybe in the 90s a little bit. In 2000 we sequenced the first draft of the 3 billion digits. At the same time that year, a child died in a viral gene therapy trial. And that's an example, that was tragic. It was not a small thing, but that really set the field back. People worked on non-viral gene therapies after the year 2000. In 2012, CRISPR happened. That was just a huge thing. 2017 to now, viral and CRISPR therapies have slowly started to get approved.
What's CRISPR?
CRISPR is a technology for editing DNA. Yeah, I don't want to get too ahead of myself, but basically the first draft of the human genome was 2000. And that was a big deal. Like, that was one of the crowning achievements of human history. How much do you think it costs to get all 3 billion digits of your DNA sequenced and emailed to you today?
$400.
Yeah, so people in the audience are going out with me, and it's like basically all I talk or think about. So yeah, it's about $400. Yeah, it takes like, you just swab your cheek, mail it in. You can just sign up with a fake name and a fake email address. Nobody will ever know it's you. And then if they sell your data, so what? It's not, you know?
What's a good company that does it?
Sequencing.com. Yeah, so there's a few companies. Sequencing.com has the best privacy policy, the best interface, just they're the best right now. One of the reasons I want to give this talk is because AI is booming, and I personally think rightly so. Genetics is just lagging, it's struggling. Like, sequencing.com is the only profitable direct-to-consumer whole genome sequencing company. And they lose money selling the kits, they only make money on the subscriptions. People should be less than 1 in 1,000 humans worldwide has been sequenced at all. We know next to nothing. Like, this is gross negligence. And it's not just for people who want to go to space or like, you know, build super soldiers or be better athletes. Countless people are suffering from diseases. Some are very obvious, some are less obvious, but many of them are influenced by genetics. We're sitting on the power to fix that. And it's under leveraged tremendously. So for $400, you can get your whole genome sequenced. Devon and I have been traveling around and buying kits with our own money. Minicircle's been buying kits for us. We've been sequencing elite athletes, sequencing elite intellectuals, and we've learned a lot. It's been awesome. So yeah, that's, let me refer my notes, make sure I'm not leaving anything out about sequencing, as simple as that is. Yeah, so the big objections are privacy. Just use a fake name. It's not, you don't have a legal obligation to give a private company your name when you sign up for genetic sequencing. Use a fake name. We know one of the guys who's high up in sequencing. He was like, yeah, just use a fake name. They don't care. So, yeah, get your genome sequenced, $400. The other one is, what am I going to do with it? So, I'm going to tell you right now. Most people I talk to are like, oh, I bet you get those sequences and dump them into AI. You know what I do? I copy and paste data from the user interface on the website, put it in Excel, and just go through it by hand. That has been by far the most productive way to analyze whole genomes. So I look at athletic stuff, cognitive stuff, longevity stuff, look at the rarest stuff in your genome, the riskiest stuff, then I make highlights from all that. This is incredibly productive and simple. It takes 30, 60 minutes. So me personally, so I'm 43, not making this up, I'm not trying to tug your heartstrings excessively, but I have an ultra rare, like 1 in 10 million mutation that puts me at a very high risk for head and neck tumors by my late 40s. I'm glad I got sequenced. So I have a number of tools to maybe prevent this or assess the actual risk I'm facing. You know, we've sequenced a few dozen people. I'd say in about 10% of people there's something really urgent and risky like this. Other people is a mix of cool stuff and different things, but most people overestimate the predictive ability of genetic sequencing data, like will it tell me if I'm going to get cancer? It doesn't really work like that. Most people underestimate the more nuanced, deeper insights that come from it. It's really remarkable, and it's really easy to go through. So at the end, I'll give you some ways to get in touch, and we can facilitate. Anybody can sequence who wants to. What's up, Bob?
I'm sorry, so you say you sort of data dump it into a spreadsheet?
Literally, by hand.
And is that spreadsheet 3 billion lines, or like what is?
No, that's a great question. Yeah, no. So they have a user interface on the website. You can, like if we look up a gene, so follistatin, if you look up follistatin, it will show you all the mutations you have in follistatin.
Okay.
Or if you look up a condition like Alzheimer's, it'll show you all the mutations you have that are at risk for Alzheimer's.
So do you have certain genes on your list that you just search for to see what's happening?
That's what's
with them?
taken a year and a half of work, is creating a curated list of conditions and genes to look up. And then I do have specialized knowledge that helps me to assess the relative importance of each mutation fast, but it's not that specialized yet.
And 2 questions, and
Yep.
I'm trying to keep it short. One is that, so when, for example, I did these online tests, I did it with 23andMe.
Yep.
Like then the sequencing that they claimed was not actual sequencing.
Yes.
It was only the SMP, so those 5 million variations. Now you have the access to the whole sequence.
Yeah.
And would you recommend doing the whole thing?
Oh my gosh, yes.
Okay.
So 23andMe, they cherry picked less than 1% of your genome. They're like, because it was hard and more expensive back then. Now, not only do they do 100% of your genome, they go back and forth over it 30 times to make sure they didn't miss anything. And it's $400. Like, this was a goal of humanity for decades, and we have it super cheap. It's like 30x coverage. You can do it anonymously. No one is aware that this exists, and if they're aware it exists, they're like, what am I going to do with it? I don't want to lose my privacy. That's part of why I'm talking to y'all tonight, is just literally, like, the more people we sequence, the more excitement there is, the more people feel safe around this, the more humanity can elevate via mastery of genetics. What's up, Eric?
So what might be some of the exciting things that we find?
Yeah, great question. What's the most exciting superpower anybody can think of that might be real?
Longevity.
What?
Longevity.
Longevity, yes. Longevity is totally major killer diseases: atherosclerosis, cancer, neurodegeneration. Atherosclerosis is super easy to fix genetically. Neurodegeneration, super easy to minimize. Cancer, more insidious, but elephants don't get cancer because they have extra copies of one gene. And then we defined most of the mechanisms of longevity between 1993 and 2005. They're actionable. Like, there's a ton of stuff that can be done there through genetics. Any other superpowers anybody would want? You can pick anything, no rules.
Healing.
Healing? That's a good one. Yeah. Healing could probably be amplified via genetics in some way. Hormone levels decrease with age, and that's definitely, most hormones are proteins, peptides. Insulin, IGF-1, growth hormone, those are protein or peptide hormones. TB500 is an analog of, I think, thymosin beta 4, which is a protein encoded by gene. We can put extra copies of it in you, feel better, no problem. Anything else?
IQ.
IQ, yes, IQ can definitely be improved through genetics. Our next product, minicircle Clotho, is one of the few genes that strongly correlated with higher IQ, so that's coming up soon. And speaking of that, so minicircle, one takeaway: X Prize, $100 million competition to treat aging effectively. We're a semifinalist. We've already progressed to the next round. We're looking for people age 50. We're looking for 50 people in Austin to sign up for our X Prize trial, which will be a combination gene therapy of follistatin and Clotho. So you get bigger muscles, less fat, cellular rejuvenation, improved cognitive function, and it's going to take place in the Bahamas. So pretty good deal overall. I'll send you some links you can follow, whatever, if you're interested in that. IQ, definitely. Anything else?
Psychic abilities.
What did you say?
Psychic abilities.
Psychic abilities, yes. This is one of the most, so there's a paper where they looked at the genetics of people who purportedly had remote viewing, and they identified one mutation in the whole genome that was associated with better remote viewing capabilities. First time Devon and I hung out, I told him about this. He was like, oh, that'd be so cool for special forces, like you flow into a room and just like, you can tell them. So I started following up on it. This has grown a lot. We're actually close to co-publishing a paper on a novel molecular model of telepathy with IONS, Institute of Noetic Sciences. It's woo, but it can get stigmatized as woo, but there's real science there. Sharks sense the electromagnetic fields of their prey remotely in the water. We have those same receptors, and that is central to the model we're looking at. So yeah, I doubt it would be like complete like mind read, but we might have some subtle abilities that are quiet that could be enhanced genetically. And this is what science is for. Science should be super fun. Like genetics shouldn't be abstract and scary because somebody's gonna sell your data. It should be like X-Men. And it should also be helping people. And that's what we're committed to driving. So yeah, telepathy and psychic ability is definitely in the mix. Anybody else? Anything else? Good?
Tell me more about your product. Is it a peptide, a protein, a chemical, a drug?
Great question. So gene therapy. That product is DNA. It's a gene therapy. It's a subcutaneous or intramuscular injection. Lasts for about a year, as opposed to a peptide or something which you have to inject daily or so. It's a DNA therapy, and specifically it's a non-viral plasmid additive therapy. I'm going to make a quick thing to explain what that means. We have non-viral, viral. And you can add function, you can silence stuff, or you can edit. So editing is CRISPR. That's like if you send some machinery into your body to edit stuff in real time, that's risky. We're probably going to get there, but that needs to be developed slowly and safely. Adding stuff is pretty easy. That's what we do at minicircle. We just add a little extra DNA into a few cells. We use falstatin as a secreted protein. So we make a little export factory in a few cells. We put extra copies of the falstatin gene into a few cells. It gives the cells the instructions to make the falstatin protein and ship it out into your bloodstream, where it acts as circulating hormone, increases lean mass, decreases body fat, and potentially improves cellular rejuvenation. Viral gene therapies use a viral shell to get the DNA into your cells in your nucleus. That is not redosable, and it can stimulate an immune reaction. Instead of a viral shell, we use a non-viral chemical method. We just use a positively charged chemical that binds the negatively charged DNA. That's how we get it into the cell. So this is right here. Our falstatin is a non-viral additive gene therapy.
So it won't be attracted to a cancer cell.
Not that I know of, no. I don't think so. I'm a scientist, not a salesperson, so when people ask me questions, I don't know the details. I give a I don't know in great detail, and I go to literature, but that's not hit our radar at all. It's not clear that even if it did, it would be particularly dangerous. Yeah, the thing is that so non-viral plasmid gene therapy is really considered safe. People just don't think it's effective. But it can be effective. It just got neglected because CRISPR blew everybody's mind and people flocked to that. So we've been developing an irrationally neglected method of gene therapy that's complementary to viral delivery and editing technologies. Basically, this is a great non-viral additive plasmid gene therapy like we do at minicircle. There's people with genetic diseases. There's a lot of people who could stand to get augmentations or improvements like now. While we work out the details of much more complicated, riskier viral delivery, CRISPR, editing, that's a great time to just boost the delivery capability of these complementary, safer, simpler, much cheaper methods. There's a number of orphan diseases we can treat. Aging is tractable with non-viral additive gene therapy. Augmentation is possible. So that's what we do at minicircle. Yeah?
You mentioned Clothos as well. Where does that fall in that?
Clotho is the same, yeah. Clotho is also a secreted protein. And same, we put it in a few cells. They act as an export factory. Clotho is effective as a circulating protein in your bloodstream. Falstatin is basically, improved body composition and cellular rejuvenation. Clotho is improved cognition and rejuvenation. Great 1-2 punch. That's what we're doing in the X Prize. Yeah. Yep.
What is you said cellular rejuvenation,
Yep.
what is the measure of that?
It's a great question. Yeah, so I'm going to be maximally responsible scientist, and I'll say we saw improved epigenetic estimates of age in our proof of concept clinical trial using follistatin gene therapy in adult humans. Epigenetic estimates of age are valuable data. I would say they're not definitive. I would say those data are consistent with cellular rejuvenation. I would say they're not, they fall short of, I would say they're consistent with cellular rejuvenation. So that's the evidence we have for that. Yeah.
And after a year, does it catch back up? Like you said, it lasts for a year.
Yeah, we just put together, so our initial trial looked at results through 3 months. There were no adverse effects. And the benefits were to body composition and epigenetic age. There were, we just put together the longer term data, 6 months through, I think, maybe 33 months was something like that. And I'd say a year is a good estimate. There's inter-individual variability. I think the serum levels of follistatin do go back down, and I would expect that the benefits probably return to baseline. I don't think there's a dip below baseline that we observe. I think that's what our data are consistent with. Yeah. A year is great because it's like long enough to be, it's longer than, injecting something every day, but it's not permanent. So it's kind of a nice sweet spot in a way. So yeah, we some of the cool mutations that exist that are harnessable with gene therapy. There are people who are much more immune to atherosclerosis and Alzheimer's. There are people with nearly unbreakable bones. There's people with superhuman metabolisms. People who can't feel pain, and that can be bad. But all these things are harnessable with genetics. Like the sky is the limit and beyond. That's just stuff that already exists in people's mutations. We can also design stuff. So I feel like I've talked for longer than I expected.
One question.
Yeah.
Affordability.
Yeah, great question.
Affordability.
So
It's gonna be affordable.
So this is medical tourism currently. Our company, minicircle, is just starting to engage the FDA to try and onshore it. Previous viral vector gene therapies were a few $100,000 via medical tourism. Our follistatin therapy is currently $25,000, which is a 10x reduction, but still very expensive for most people. The more this catches on, the more it can drive the price down. But that's where it's at right now. So we achieved a 10x reduction. We want to drive it even lower, make it more accessible.
Friend just spent $1 million on his cancer. Everything.
Do you know, was it a gene therapy or just another?
It wasn't.
Yeah. Yeah.
It was $1 million.
Yeah. It's there's a lot of money.
He had four protocols.
Yeah.
He qualified for the last protocol. Got irradiated. Was in a what do you call it? Sterile chamber.
Gotcha.
For
Yeah.
quite a long time, and
There's a lot of money, out there that people want to be healthier, and there's a lot of money floating around out there. A lot of it is not optimally targeted. Like, so yeah, we're going to spread awareness here, spread knowledge. Yeah, because this should be accessible financially. It should be accessible in terms of people's understanding. We're very mission driven. minicircle, Devon and I are very mission driven, and that's our mission, to make genetics accessible to everybody, so we can all, be elevated. What's up, Eric?
2 questions. Do you see that price going down? I mean, I know it's gone down from 200K to 25, which is significant. Do you see it going down further?
Yeah, I do. I mean, so we had we have been developing a luteinizing hormone gene therapy, which would, be a testosterone replacement type of thing. And I think we're planning to market that for maybe $500. I'm not on the marketing end of things, but it's possible to sell it for less. And, we're still a startup with one product currently. Yeah, the price can come down further for sure.
Okay. Okay. And then you mentioned a year. Is there a certain point where it's gonna be, you see it being like, take one shot for 5 years or 10 years?
Yeah.
Could last.
Yeah, I think with non-viral, it's probably going to be about a year. Viral and CRISPR can, I think, last longer, but they're also riskier. So I'd say right now we're looking at about a year with non-viral. In the future, longer and even permanent. You can edit and permanently change the DNA. Yep. Any other questions or thoughts?
Yeah, just in terms of Project Limitless, like, what would you consider mission accomplished? Like, what is your sort of dream target?
I think Devon beating Levon would be
Yes.
mission number one. I think it's, I think it's very fluid. I think I feel like we both have a sense. It's fluid. It's a very alive project. I think spreading awareness and excitement, getting more people sequenced safely, and just creating a wave of development around genetics that doesn't get co-opted. People's data don't get sold. Stuff gets developed faster in a way that's accessible to just, to get humanity on board with the unifying mission to get us towards the stuff we all really want. Science is collaborative. So yeah, getting people sequenced, developing gene therapies, treat people with disease, elevate humanity towards a proper spacefaring civilization, unifying mission, those are the goals of Project Limitless. Yeah. Great. Any other questions or thoughts? I'll throw up a few social media things and so on and so forth. Thank you for all your questions. Also, thank you everybody for coming out. There's some minicircle swag over there on the thing. I'll throw up some social media stuff. Yeah, thank you all very much.